and thinking about the idea of, how do we look at the process? What is the process of the evolution? How do we kind of study that a little bit? And then some unique things you have to think about with regard to microbes that you probably don't hear too much in like our standard evolution class that you take here. So, it's always fun, with my expertise in microbiology, if I talk, like I would say I'm a microbial ecologist. And so if I talk to a macro ecologist, really different conversations sometimes when it comes to these ideas. And hopefully by the end of today, you'll get an inkling of what I'm talking about here. So in general, I think that you've all been exposed to this idea that when you look at the evolutionary process, we're thinking about mutations as part of that process, which is true in the microbial world as well. And what is that? It's just a change in the nucleotide sequence. All right, so we're looking at the nucleotide sequence, DNA, but with regard to viruses, we have to, which we'll learn about, well, COVID, it's an RNA virus. So, so nucleotide sequence is a little bit better way to say it because it could be DNA, and viruses, it could be RNA. Okay, so a change or some kind of event happens. You can force the mutation, like in a research lab. Sometimes when we work with microbes and we want them to mutate, we might hit them with ultraviolet light. You'll actually work with ultraviolet light later in the semester and see what that does to microbes. And you could apply certain chemicals that we know are mutagens and will mutate the microbes. And then I say, and other factors, because there are other things going on that can cause mutations, which I'll get into. And just remember, adaptive, when someone says that's an adaptive mutation, what we mean is that it improves the fitness of an organism, increasing its survival in its current habitat. In its current habitat. You have to keep that in mind. Put it into context of in its current habitat, because if that microbe's habitat changes, their adaptive mutation may become not adaptive anymore and may do them in. Okay, so you have to put it into context of what is their living environment like. Another thing to keep in mind is that prokaryotic cell types, so bacteria and archaea, they are genetically haploid, which means they just have one copy of each gene. We have two. We got one from our mom and one from our dad. All right. So we have 23 chromosomes from our mom, 23 from our dad. Microbes don't do that, right? They are genetically haploid. So that raises some interesting situations for them. Now, having said that, we now know that there's a process called gene duplication, which I'm not going to get into because I'm not a geneticist. So I probably don't, I know some of it. I just don't know the nitty gritty details. But basically, when they're reproducing themselves and going through that process, they can duplicate one of their genes. And then sometimes it stays as is, but we've also seen that some of these genes, it's like the microbes tinkering with it, like, oh, I got an extra copy. What can I do with it now? And they start tinkering with it over time, and they can mutate it further to become something like unique and new. And so that's kind of an interesting process. There's scientists who will study that in the lab and watch these microbes kind of, I like to think of it that they're playing around or tinkering with it. We'll talk quite a bit about this horizontal gene transfer. I'll introduce it right now and we'll get into it later. Horizontal gene transfer. So now we're talking about this idea that two living microbes hanging out together can share DNA with each other, all right? Horizontal. And so you have to have coexisting microbes hanging out with each other, and they can transfer genes. Now, this was a huge deal when this first was discovered. We'll talk more about it. We're going to look at the different ways microbes do this because they have figured out several different mechanisms. And then don't forget, you can see gene loss occurring in microbes at a much higher rate than what you see in animal cells, in animals in general, even single-celled eukaryotic So when is this an advantage? If you're a parasite, if you are an obligate symbiont, you can imagine. So let's say you're hanging out and like in a endosymbiosis idea, for example, it would kind of make sense to purge away some of the genes that your host is providing that for you anyways. So why should I do it for myself when my host is doing it for me? And so you might purge those. And microbes are bacteria. They're really quite good at like, yeah, I don't need this anymore. I'm going to purge it. And so that is something that often gets forgotten when you talk about evolution, that, with higher organisms. So we'll come back to that idea. All right, so phylogeny. You often hear this term. We're looking at how the history of those organisms in relationship to other organisms. And this is usually done through nucleotide sequence data that we compare in order to put together, a so-called phylogenetic tree. In order to do that, we use some kind of chronometer or a molecular clock to compare across different organisms. And so you have to pick something that all organisms have, and it's being used the same way amongst all those organisms. And hopefully isn't changing too fast, but does change. It's just not too fast in terms of mutation. All right, and so this was some of the work that Dr. Woese did, Carl Woese, in the microbiology realm. He was able to think through this problem and come up with the idea that we should use the small subunit ribosomal RNA genes. We should focus on that nucleotide sequence that codes for the small subunit ribosomal RNA, which is being shown here, which is, it's decent size. And the reason Carl Woese was proposing this is that it's found in all living organisms, all domains of life. It's slightly different when you look at prokaryotes versus eukaryotes. They're different sizes. So the ribosomal RNA in prokaryotes are the so-called 16S, referring to their size, 18S for the eukaryotic cell type. So we have 18S, but bacteria have the 16S. But they all are functionally doing the same thing. Okay, they're functionally constant, and they're conserved, which just basically means they don't change a whole lot. It takes a while, and it's big enough. And so Dr. Woese proposed, this is maybe a stretch of DNA that all organisms, code for this. Let's use this and compare it amongst all the different species and see how it maps out on the phylogenetic tree. So that's kind of the gold standard right now in terms of generating that output, the phylogenetic trees. And it's gotten to be pretty mundane. You can send your samples off and have this done quite readily. And basically you just get that section of the gene targeted, and then you amplify it, and then you sequence it, and then you look at the output. And so there's companies out there that will do this for you. And then when you ask them to, they'll put it into context of a phylogenetic tree. And in the microbial world, what we say is that if it's 95% or greater similarity when you compare the sequencing, then we say it's the same species. Okay, 95. So I do want you to remember that number. 95%. 95% or greater for the 16S. That's the microbial, bacterial and archaea. All right. So when Dr. Woese posed this and then started doing this kind of research and looking at the output, that's when we started to see that you can't lump archaea and bacteria into the same domain. They belong in their own domain. And so that's when we decided to, well, not me, but they decided to split. And they're like, yeah, the archaea need their own domain. And then you have the domain bacteria, and then the domain archaea. And so it was a very powerful technique that's still used today. And nothing really has come to surpass it so far. There may be something down the line, but not yet. So this is an example of a phylogenetic tree. I can't get the red dot to circle the right thing. It wouldn't let me the way it came, but anyways, let me just, so you have down here the LUCA, last universal common ancestor. And then you see your phylogenetic tree, the speciation off of that. And so you can see the bacteria over to the left, archaea in brown in the middle, and then the eukarya in the gold off to the right. So you need to know the 3 domains of life: bacteria, archaea, eukarya. Then this is a partial tree. This certainly is not the whole thing. This is just giving you some examples, but you can see some, generally when you see the words, those are phyla for the most part. There's a couple of exceptions. For example, you see right here, chloroplast. I think that's kind of funny that they put that there like that. So again, remember, what we can now call chloroplasts, they were once free-living ancient organisms. And so they actually think they know exactly where, that organism, what it was before it got engulfed and became the chloroplast. Mitochondria is on here too. All right, so remember, mitochondria were once free-living organisms. And so they're on there. But then you can see like proteobacteria. Well, we don't think a lot of phylum names. Mostly, yeah, a lot of these are. Okay, let me point out a couple here. Euryarchaeota, that word is a phylum name. And then Crenarchaeota, that's a phylum name. So right now, there's only 2 phyla of archaea that we know of. There used to be 3, and then with further sequencing, they said, no, there's really only 2. So I would like you to recognize those 2 words, please, and know that those are the phyla of archaea currently, Crenarchaeota and Euryarchaeota. As far as the different phyla of bacteria, there's a lot, so I'm not going to ask you to memorize all of those, all right? Lots of different phyla that belong into bacteria, and then same with the eukarya. So another thing to pick up on with this phylogenetic tree that's easy to see, and once you understand the rule here. So let's look at what's coming off the LUCA, off to the right, to the eukarya line. Do you see how the archaea are coming off that line, right? They're coming off here. So what that is saying is that the archaea are more closely related to the eukarya domain than they are to the bacteria domain, which was a huge surprise. That was not what we thought. We used to lump archaea and bacteria all together and said they're all the same thing. And then Woese said, well, let's do the 16S ribosomal RNA. And now it's actually greatly shifted our idea, our understanding. The archaea are actually more closely related to the eukarya because they're coming off that line from the LUCA. Okay, so that's something to think about. And I've showed you this one already as well, with the idea of the, I'm just trying to edit this a little bit. It won't let me edit. The phylogenetic tree, where you see most of the life on the planet, is in the domain bacteria and archaea. So super abundant, even though most of them we cannot see with the naked eye. I would have loved to have been a fly on the wall back in the day when, Louis Pasteur and Dr. Lister had to convince people that there's these little wee beasties that you can't see with the naked eye, and they're making you sick. I mean, back in the day, surgeons would never wash their hands in between patients. They didn't understand that there was germs on their hands they were transferring. And in fact, the dirtier their hands and uniforms were, they kind of took pride in it. It was almost like I've done so many operations, look how messy I am type thing. And when they tried to get across the point about germs and you need to be cleanly and wash your hands and the hospital ward should be clean, it met with a ton of resistance and ridicule. In fact, didn't Lister go crazy? That's probably not the right way to say it, but he ended up getting depressed, severely depressed, and ended up in an asylum because he was ridiculed and they basically made him a laughing stock, and his career suffered And in the end, he was vindicated. What he said was true. But during that time point, it did not go over well. So wee beasties that we can't see. In lab, you're going to be doing a lot of this idea, where you're going to be looking at microbes, not really sequencing them. That's expensive still for doing labs. You'll do more of these types of things, where you're going to be trying to separate out and identify who's who based on, like, what do they look like when they're growing across an outer plate? What kind of physiology do they have? How do they behave when given certain nutrients? That would be a way to define who they are as well. Are they mobile or not? Do they use oxygen or not? So there's a lot of other ways besides DNA sequencing or nucleotide sequencing that you can get a feel for the microbe. And so I call this getting to know your microbe. You're going to be doing that a lot. And some of the things listed here, like cell wall chemistry, is not something we'll do. That would be some pretty sophisticated chemistry that you have to do, but it's doable. Lipid chemistry, when I was a postdoc, we would use that to compare microbes within the same species. Within the same genera, sorry. That's kind of a handy tool. So you're going to get used to some of these ideas in the lab for looking at similarities and differences. But when you think about microbes, there's a problem in the sense that if you think about Charles Darwin's idea of the origin of the species and the idea of natural selection and evolution, he introduces the idea of the biological species concept, which is that species are part of a reproductive community. And if they can no longer produce viable offspring, then we start thinking they're different from each other. So, Homo sapiens, we can produce viable offspring with other Homo sapiens. We're individuals within that species. But so, okay, that makes sense at the macro level. You can see this going on with sexual reproduction. But what do you do when there's organisms out there that really don't have sex? Then what do you do? This is where it starts to all get really weird. And when I talk to macroecologists, they kind of get about this. And it's like, well, in the microbial world, microbes don't have sex like animals do. Like, we have sperm cells and egg cells, which have half the number of chromosomes compared to the rest of the body. They combine. That's the idea of meiosis, to produce those cells, and then you get fertilization. Microbes don't do that. So now it's like, okay, so how do we think about this? They don't have egg and sperm and don't go through sexual reproduction. How do we do this at the microbe level? So the second bullet point was proposed by Lynn Margulis and her son Dorian Fagan back in 1986. They said maybe in the microbial world, this is how we should think about their version of sex. We can say that it's formation of individual organisms that contain genes from more than a single source or parent. So I would like you to know that definition because it's different than this idea of meiosis producing egg and sperm and then getting fertilization to occur. So this definition, it works. It serves a good purpose, and it's a framework to consider, I guess, the microbial version of sex. All right, so this is like their version. And so it's formation of individual organisms that contain genes from more than a single source or parent. So now we're looking at this idea of horizontal gene transfer, where we can see two microbes hanging out with each other and they may share genes with each other, but they don't have egg and sperm. That's not what's happening. But it's their version of sex. Okay. So what they have found is that, initially when they were looking at some of these ideas in the lab, when they were doing research, they would work like with individuals of the same species, so like 2 E. coli cells that they'd grow in a broth. And then they'd watch to see what would happen. And then they'd sequence them. And they could see that there was horizontal transfer going on. And that kind of was mind-blowing. That was such a out there concept. We just had no idea that microbes could do that. So they said, yeah, look at this. Microbes can share genes with each other like 2D cells and do this. And so that was a big wow moment when that got published. And they're like, wow, look at this. You know, they're sharing genes. Well, then other scientists came along and said, oh, yeah, well, there's even different species of bacteria that can share. So that would be like Pseudomonas aeruginosa is one species. Pseudomonas fluorescens is a different species. Both Pseudomonas, but they're different species. And they were sharing genes. That was even more of a mind-blowing thing. Like, wait a minute, they're not even that related, and they're sharing genes. And on the story goes. Now we know that microbes share genes in different genera of bacteria, for example, and from organisms even. So now we're saying, hey, guess what? Like there are certain bacteria that share genes with plants, for example. And then the plant takes that DNA and expresses it. There are animals that will interact with their microbes that are coexisting with them that will do this as well, including humans. So it is kind of mind-blowing to think that you have this kind of horizontal gene transfer going on, and it's been going on, I'm sure, for billions of years, and we never picked up on it until fairly recently. So they often use these terms. They're promiscuous cosmopolitans because they're quite good at it. There's certain methods that they'll employ to get this to happen, and it's pretty efficient and fairly common across the different microbes that have been studied. So they've been taking lots of environmental isolates. They take microbes that hang out with humans and other animals, and they look at this, and more often than not, the answer is, yep, they're doing it. All right, so then they started saying the promiscuity of the microbes, that they're able to do this. So just to clarify, we're talking about two coexisting microbes living together in a habitat, and they're getting genes from each other somehow. Vertical, on the other hand, is like the true sex idea back in the animal world, where you have the transfer of entire genome from parent to offspring. So you're a result of vertical gene transfer when you were born. When a bacteria decides to go through cell division, its progeny cells vertical, then that's vertical gene transfer. Okay, so microbes can do vertical. It's when, like an E. coli says, yeah, it's time to divide, and then two progeny cells are made, it will have transferred the entire genome into those two cells. Okay, so in microbiology, it's a little different because there's no sex in the sense of meiosis and sperm and egg and fertilization. So when we think about speciation in the realm of microbes, and bacteria have been the most heavily studied type of prokaryote, we see that, yes, you can have natural selection occurring. We see that happen, where, remember, natural selection is repeated periodic selection for a favorable trait. So the habitat is influencing and affecting which genes the bacteria keep and express, which ones they make turn off, which ones they might purge. All right. And through that process, you can get speciation to occur if it's dramatic. So that's certainly still happening. Natural selection is happening, but don't forget, in the microbial world, horizontal gene flow or gene transfer is very important. In fact, if you talk to an evolutionary microbiologist, they would argue that the bottom image is more appropriate for what's really happening in our world compared to the top image. So usually when you see, when they talk about evolution on Earth, they usually draw something like that top image, where you have like this last universal common ancestor and everything comes off of that. Now they're saying the more they learn about horizontal gene transfer, they're saying, oh boy, we need to think about all kinds of horizontal gene transfer going on over time from the last years. So it's much more complicated than what we originally thought now that we know more about this horizontal gene transfer. So anyone who's taken the evolution class here recently, do they even talk about this idea? No, remember? Maybe a little. So it's still not commonly, I get the feeling anyways, it's still not commonly talked about in this way in a typical evolution class. But if you talk to an evolutionary microbiologist, oh yeah, this is what's really happening. So I think there's still a disconnect that is going on a little bit between the micro and the macro people. This is from another resource, not your book. And again, this scientist was also, saying we need to think about this horizontal gene transfer, which is really influencing and driving a lot of the evolutionary processes going on Earth. And I think that's really fun to think about. And then I like this image too, because this image of this is a kind of a pared down kind of a tree idea. But I like how they're talking in this idea that there's gain of genes and loss. They include that as well. Okay? I think that's important to think about gain and loss, especially at the microbial, in that realm. Microbes are really quite agile, adept at like, yeah, I'll take this gene and incorporate it. And you know what? I don't need this one. And they'll purge it. They're just much quicker and agile at doing that compared to the typical eukaryotic cell. So a lot of obligate symbionts, including obligate pathogens that need their host, a lot of them have lost key stretches of their DNA. And if they're separated from their host because they've lost so many stretches of DNA, they can't survive. They're obligate. They have to be with the host, for good or bad. So as far as the idea of what Dr. Woese was proposing with the let's sequence the ribosomal RNA and let's get at the the relatedness. So when I showed you that one image that the bacteria and the archaea are actually further separated than we thought, and that the archaea are more closely related to eukaryotes. These are some of the things that you can say are similar or different. I think this one is from your textbook. Might be slightly different table number based on the new edition, but this is good for study purposes. But let me show you this one, which I'm not sure, this one's not in your textbook, but it's a little easier for me to tell you what I want you to study. And so I'd like you to be able to think about these three domains of life with some key aspects. And so let's go start at the top with prokaryotic cell structure. See that the archaea is prokaryotic. But let's go all the way down. Well, that first, all the way to the red bar. All right, so I'd like you to know this first section, the morphology, all the way down to this red bar. So be able to compare those three domains of life with regard to different criteria. And we'll be talking about criteria through the semester. Fairly soon that we get into cell structure and function. So I'll be hitting on a lot of these anyways, but this is kind of a one-stop shopping where you can see that nice comparison between the three domains of life. Okay. Huh. It's weird. All right, there we go. Finally. All right, so there's a few slides here on the end of this slide set, all the white ones. Okay, I'd like you to use these as study tools to accompany the reading in the textbook. So this is Dr. Lenski's grand lab experiment, and professor at MSU, I believe. In fact, one of our former grad students worked in his lab when he was taking. He came up to Northern to do his grad work, and he was taking microbial ecology that semester with me. And I started talking about this in my microbial ecology. He's like, "Hey, I worked with him. I did this experiment." So it was really cool to see, hear firsthand what they were having going on in this experiment. But basically, Dr. Lenski's group wanted to see if they could, could we see evolution in action over, a space of time while they're studying these microbes. And basically what they were doing is growing E. coli, and every day, including, Christmas and holidays, everything, every day they would come in and subculture into fresh media. They take a subset and put it into fresh media, let it grow, take a subset, put it into fresh media. And they kept doing that. And then they take samples over time and freeze them and then sequence them. And then they'd look to see if there are changes going on over time with this E. coli. And so basically they took for a lot, and then they would plate them out. Where's the picture? Here's a, oh, yeah. My computer's freaking out on me. Go away. All right, let's try now. Do you see, those are all Petri plates behind the students sitting cross-legged, like a pyramid of Petri plates. So they're trying to show the scope of the work being put into this experiment. It's gone on for years and years. And I'd like you to read up on what they discovered. It's a pretty interesting discovery. And so when I talk to people, whether they're biologists or friends at a party or whatever, and the topic of evolution comes into the discussion, I can easily give them examples of evolution happening in our lifetime by talking about what microbes do. And so it is very easy to show that happening in the lab, that natural selection, adaptive fitness, it happens on a regular basis within our timeline. So we don't have to wait for our ancestors to figure it out. All right, so please read through that section and use these slides as a study tool for that. Kind of fun to think about. Okay, that's it for that slide set. Did I put the next one in? I had a feeling that was going to happen. All right, let me get into stop share. Ah, all right. All right, so now it should be posted, gene transfer. So this is continuing on with this idea of horizontal gene transfer that we just talked about. So now I'd like to get into more details on it. So when they first discovered that this was happening, they first published on, they do it this way. And then other scientists came along and said, but wait, they can also do it a different way. And then, so there's actually like 3 main ways that we know of. Well, actually there's more than that, which have just recently been discovered. So let me get into it will let me. Oh my goodness. Oh, I have to discard. Why does it show up there and not on my laptop? I don't know. But it does. Okay. Gene transfer. Okay. So this idea of horizontal gene transfer and mutations and adaptive selection. So let's look at the left. This is a colony. Well, this is a Petri plate. And what they did, they took a agar plate, and you're going to learn this tomorrow in lab, to do a spread plate exercise where they pipetted a suspension of microbes on the plate, and then they used a glass spreader to spread it all around. And then what they did was, in the center of this Petri plate, they put antibiotic disc, kind of see it, that it's like a filter paper disc that's impregnated with antibiotic. They put that on after they spread the bacteria. They put that on, and then they incubated the plates. And then after a couple of days, they took them out, and this is the result. So what you're seeing is what's that antibiotic in that paper filter disc. It over those 2 days of incubation, that antibiotic diffused out into the agar. And any cells that were spread there initially that were sensitive to that antibiotic were killed off. All this gray further out, those are the microbes that weren't impacted by the antibiotic. The antibiotic didn't make it that far. And so you're seeing lots of growth further out, but you see this kill zone or inhibition zone where very little has grown, except I think you can see even in the back, can you see those colonies, those dots within the kill zone? So that, the first time someone noticed that, actually had the attention to detail, and went, what's that? That was a big aha moment, because that is showing that there was a cell that landed on that agar plate when they initially set up the experiment that must have been resistant to that antibiotic and was still able to survive and grow and divide until 2 days later it pops into a colony that we can see with the naked eye. And so it looks like there's a handful of colonies. All of these colonies resulted from one cell that was resistant to the antibiotic and managed to keep on growing. And so that was, again, another big moment in science that someone paid attention to that, went, okay, how come that cell was able to survive and all the millions of other cells died off? And so we start thinking about this idea of, I think most of us are aware of antibiotic resistance is a problem, right? It's a big problem. And I'm sorry to say, for your generation, it's going to be even bigger than my generation. And so unless something big happens soon, which I hope will, and it's encouraging, there's some hopeful things going on, but it's scary. Like when I go to conferences and I talk to medical microbiologists that study antibiotic resistance, like, ooh, time to go have a beer after that one. You know, it's just, it's an intense conversation, and it's kind of scary. So antibiotic resistance is a big deal, and I'll be talking a little bit more about that in this class. So how did these cells become resistant? Well, they saw this phenomenon. Now the question is, how did they do this? We need to understand this. We have to understand it so we can limit it in the case of antibiotics. One method that can be employed is called transformation. So you need to know that term. Transformation is actually free DNA. They actually use the word naked. Naked DNA, what that just means is that it's DNA that has spilled out of a cell that died. When cells die, they tend to break open or lyse. That's the fancy word. They lyse open. The inner contents spew out, including the DNA. And then a nearby microbe might go, ooh, there's DNA here. I'll take some up, right? So transformation it's usually fragments of DNA, because DNA is huge. You can't take in the whole molecule. So they'll take in segments, and it's usually around 10 kilobyte base pairs, 10 kilobase pairs that they can take up in one chunk, and they'll bring it into their cells. And so this was discovered fairly early on. Some of you may, I don't know if they still do this. I think they still do this in BI 218, Intro to Cell and Molecular. And so this is something that, kind of routinely, we know how to get certain microbes to do this. And it's not too painful of a lab experiment to do this. And so they demonstrated first that, of course, okay, we can show that it's happening in a lab if we force the situation and create, these conditions. And then, of course, they asked the question, do real microbes do that out in the real world? And the answer is yes, they do. And so it's not just this artifact of, creating perfect conditions in the lab. However, not all microbes are necessarily have been witnessed to do this. And I'm guessing that's maybe just because of our lack of imagination. We just haven't figured them out yet. But anyway, currently they say not all cells are transformable. And so we'll stick with that idea. That's what they're saying. And so they know that there needs to be certain genes present in that microbe. They need to be turned on at the right time. And so they start talking about this word competent. So if they have those genes, then we say they're competent. That means they're capable of doing this transformation process. And so one of the concepts of this are these genes that code for special proteins. And these proteins have been called transformosomes, which I guess that's a pretty good name, transformosomes. They essentially are proteins that get made and built, and then they form this complex that allows for this DNA to enter the cell. I'll show you a picture. And so it's basically just a mechanism of entry so that it's a way for these sections of DNA to get brought into the cell. They're also finding out that it seems that this process called quorum sensing is necessary in especially gram-positive bacteria. Not universal so far, but it seems like it's often a strategy. And I'll talk more about quorum sensing, but basically quorum sensing is a way that microbes will assess who's around them, like which microbes are around me, including which of my own species. Are there other ones out there? And then based on that information, they may decide to behave differently. And it really is a super useful mechanism for microbes because then they can change what they're doing and take advantage of certain situations that will be beneficial for them. I'll talk more about that. So in the lab, they've done a lot of study with Bacillus subtilis is their kind of guinea pig for doing a lot of the transformosome with the help of quorum sensing idea. So that was the model bacteria used in a lot of this research. So they know it can happen. They know that bacteria, especially B. subtilis, can take up small chunks of DNA. And then what they do is they'll incorporate that into their own DNA, and then they can express it and make use of it. And transformosomes need to be built to get it to happen. And so, but of course, that begs the question, why? Why are they doing this? It's a lot of work. You got to build those transformosomes. You got to do quorum sensing. That's some work to do as well. But if you think about it for a minute, well, some of that naked DNA could be useful. It could have genes on those stretches they take up that they could actually even use for food. They could break down the nucleotide sequences because that has yummy stuff that they need. They could use it for nutrition, a source of nitrogen, for example, and carbon. Maybe. Maybe they do it to repair some damage. Remember, they're haploid. These are haploid organisms. So if they can take in let's say one of their genes got damaged, if they can pick up a new piece and it's like, oh, this one's fully functioning, I'll just use this one instead. And so you could see that might definitely be an advantage. And then this last one too, it could be just a way for them to acquire a new gene that they've never had before. And they're like, ooh, this is good. This gives me an advantage in my current habitat. I'm keeping this one. And I'll hang on to it. I'll put it into my DNA. All right. And so, of the 3 bullet points here, DNA as food is probably the least used reason. That's probably not such a thing, but the repair and acquiring new genes, you could definitely see where that could be a huge advantage. So this is not a molecular biology class, but I just wanted to point out this process of transformation in a gram-positive bacteria. So we said they do quorum sensing. So they're actually showing quorum sensing here. I'm not going to get into it right now, but basically the feedback that you see is part of the quorum process, and it's essentially what it is, signal cascade system, which I'll talk about. And so that kicks off first, and then it sets the stage for shifting gene expression to build the transformosome complex. And once that gets built, then the naked DNA can be brought in. Okay, so kind of like a 1-2. First quorum sense, if everything's good to go, shift your gene expression, make those proteins, get your transformosome built, then take in your DNA. So you can see this is kind of a, this is work. This is energy and resources that are being put into it. So there better be a payoff, right? Otherwise, it's a lost effort. But if everything goes okay, they can bring in sections of DNA. Hopefully that can be super useful. Okay, so in gram-positives, we tend to see this. And so you'll learn about gram-positive bacteria, by the way, next week in lab. So we talk about gram-positive bacteria and gram-negative bacteria. It's two huge groups of them. Okay, we'll stop there and continue on with gene transfer on Wednesday. Oh, I forgot to hit share. So the second half of the recording isn't going to work. You're not going to see it. You'll hear it, not see it.